During mitosis, cells undergo a series of dramatic shape changes, rounding up in pro-metaphase, elongating in anaphase in preparation for cytokinesis, and flattening back out upon mitotic exit. Sébastien Carréno from the University of Montreal has previously found that the ERM protein Moesin, which increases cortical rigidity by linking actin filaments to the plasma membrane, is important for this mitotic remodeling.
In 2008, when I was a postdoc, I have found that Moesin was important for cortical stability during cell division and for cell rounding at metaphase entry. Then after, when I started my lab, we decided to study how Moesin was regulated during cell division.
Moesin is the only member of the ERM family present in Drosophila. Carréno and colleagues, led by graduate student Chantal Roubinet, began by examining the localization of GFP-tagged Moesin over the course of the cell cycle.
In this cell type, Moesin is mainly in the cytoplasm during interphase. Then, when cells enter prophase and metaphase, Moesin is recruited everywhere at the cortex, and this is related to its function in a cell rounding. Then, Moesin accumulates at the equator. There's some remaining Moesin at the polar cortex in anaphase, but it's mainly localized at the equatorial plane and at the cleavage furrows. And then when cells re-enter interphase, Moesin is inactivated and is lost from the cortex.
Moesin's localization to the cell cortex is promoted by a kinase called SLIC, which phosphorylates Moesin on a conserved threonine residue. A Moesin mutant mimicking this phosphorylation is constitutively active and remained associated with the entire cortex throughout the cell cycle. Cells expressing this mutant failed to properly elongate during anaphase, often resulting in defective cytokinesis. Anaphase elongation requires contraction of the cell equator and the simultaneous relaxation of the cortex at cell poles. Normally, the reduction of Moesin and actin at the polar cortex allows anaphase cells to relax by forming transient membrane blebs at their poles.
What we postulate is that these blebs that occur in anaphase at the polar cortex release the pressure that is done by the furrowing of the cleavage furrow, and you have to imagine a balloon. The balloon will explode if you press it too much, and here in the cell what happens is that this pressure is released by the formation of the bleb.
Cells expressing constitutively active Moesin probably fail to elongate because the mutant remains associated with the polar cortex, recruiting actin to maintain the stiffness of cell poles. Yet anaphase cell morphology is also affected if too little Moesin and actin associate with the polar cortex. Moesin was found to transiently accumulate in blebs, along with actin and myosin, just before the blebs retracted back into shape. Cells lacking Moesin, or its activating kinase SLIC, fail to recruit actin and myosin to resolve their blebs, leading to uncontrolled bleb growth and excessive relaxation of the polar cortex.
You need to have less Moesin at the polar cortex than at the equator in order for the cell to elongate. But what is also important is to retract these blebs, and then Moesin comes back.
This suggests that Moesin's localization and activity are tightly regulated. Roubinet et al. Decided to look for a phosphatase that might oppose the activation of Moesin by the SLIC kinase.
Here we took advantage of Drosophila cells where the genome is less complex. We knocked down every catalytic subunit of phosphatases, and we found that only one catalytic subunit, when depleted, led to overactivation, overphosphorylation of Moesin.
In the absence of this phosphatase, known as PP187B, Moesin localized all around the cortex throughout the cell cycle, even in anaphase, resulting in cytokinesis defects similar to cells expressing constitutively active Moesin. However, Moesin still showed some preferential enrichment at the equator of anaphase cells lacking PP187B.
So we thought that there was another mechanism that will reinforce this polarity of localization of Moesin during anaphase elongation. Naturally, we went to the PIP2 because this phosphoinositide has been shown previously to regulate ERM proteins.
Binding to phosphatidylinositol 4,5-bisphosphate activates ERM proteins like Moesin. Roubinet et al. Found that this phosphoinositide was enriched at the equator of anaphase cells, and that rapidly reducing PIP2 by inducing the membrane localization of a phosphoinositide phosphatase caused the immediate loss of Moesin from the cell cortex and excessive polar blebbing, similar to cells lacking Moesin. The researchers then looked for the enzymes that might naturally modulate PIP2 levels at the mitotic cell cortex.
In Drosophila, you have 31 enzymes that could, at one point, modify the level of PIP2. So, in fact, we did this candidate screen by time lapse, so that was hard work, but it was pretty good because we identified 3 enzymes that, when depleted, trigger this anarchic blebbing during anaphase. A PI4-5 kinase Skittles, the well-known P10 phosphatase that makes PIP2 out of PIP3, and a PI4 kinase that we study in the paper.
Skittles and P10 both accumulated at the cell equator in anaphase, where they contribute to PIP2 production. Moesin was lost from the mitotic cell cortex in the absence of Skittles, resulting in excess polar blebbing.
So, there are at least 2 things that are important concerning Moesin activation and localization during cell division. The first one is that you need to turn up Moesin at pro-metaphase entry in order to have the rounding of the cell, and you need to turn off Moesin at mitosis exit in order for the cell to enter into interphase. We think that this is mainly controlled by the SLIC kinase and PP187B phosphatase switch, and the other important fact is that Moesin has to be enriched at the equator and less associated with the polar cortex in anaphase in order for the cell to elongate. We think that PIP2 plays a major role here because PIP2 accumulates at the cleavage furrow due to the action of Skittles and P10, and by accumulating at the cleavage furrow, create a polarity of localization of Moesin.
Carréno and colleagues are now investigating how the proteins controlling PIP2 synthesis and Moesin phosphorylation are regulated over the course of the cell cycle. But in the meantime, you can learn more about the regulation of Moesin and its contribution to mitotic cell shape changes in the paper by Roubinet et al., published in the October 3, 2011 issue of the Journal of Cell Biology.